This lecture is going to be a lot shorter than the last one. I thought we had done pulmonary and systemic circulation. Maybe the PowerPoint's just really messed up. We're going to do that. And then we're going to go into the valves again, and although we've already learned about valves, and then we're going to talk about some cardiac histology. Okay, here's the heart. And remember, you've got the right side of the heart and the left side of the heart. And the right side is basically, we've got all these capillaries in the body, in the tissues. They're microscopic. Say this is in a skeletal muscle cell in your thigh. That's this capillary bed is around that muscle, for instance. Well, it's receiving blood from the left ventricle. Let's separate this here. The left ventricle is pumping out high oxygen, low carbon dioxide blood to the tissues of the body. Here we are talking about the body systems, so it's systemic circulation. I hate it when it does that. Anyway, this is high oxygen, low carbon dioxide blood, and when it gets into the tissues, whatever tissue that this capillary bed is in, the unit of blood is only going to go through one capillary bed. It doesn't go from one capillary bed to the next capillary bed to the next capillary bed. So each unit of blood is different, is destined for a different capillary bed in the body. So here's this capillary bed. It's in the tissues. And what's going to happen is oxygen is going to leave the blood and go into the tissues. And because the tissues are constantly producing carbon dioxide by that process of aerobic cellular aerobic respiration, we've got this mitochondria, and it's going to make ATP. And to make ATP, we have the requirements of glucose. There's my cat. And oxygen. So we need oxygen for the tissues to make ATP, and we also need glucose to be able to get into the cells. And then the mitochondria, we can make ATP other ways, but I'm just doing a quick version of this. Another waste product is water and carbon dioxide. So we're always producing carbon dioxide in our cells. Because we have more carbon dioxide in the cells than out, that's why the carbon dioxide leaves the cells and will enter into the blood. So we've got this unit of blood going back to the right atrium. We started with the left ventricle. And over here, we've got low oxygen, high carbon dioxide blood. I'm going to pause to let the cat out because otherwise we will have no peace. Okay, so that low oxygen, high carbon dioxide blood is going to go to the right atrium, and then it will go into the right ventricle. And if you recall from the last lecture, when the right ventricle contracts, it sends blood to the capillary beds of the lungs. So here, down here, we're talking about all the body tissues, and up here, this is called pulmonary circulation. Pulmonary circulation, because we're sending that low oxygen, notice we're the same blood here, high carbon dioxide blood to the lungs. And what's going to happen in the capillary beds of the lungs is that carbon dioxide will go into the capillaries of the lungs and then pass into the alveoli, and then we'll breathe out the carbon dioxide, and then we're breathing in oxygen, so oxygen will go from our lungs into this capillary bed. So basically, carbon dioxide is going out, and we're exhaling the carbon dioxide, and we're inhaling oxygen, and then it goes into the blood from the alveoli of the lungs. Now, this high oxygen, low carbon dioxide blood is destined to this guy, to the left atrium. And this is the high oxygen, low carbon dioxide blood that will go from the left atrium down to the left ventricle, will be pumped out of the aorta to the body again. So these are the two primary circulations of the body. We did coronary circulation in the last lecture. If you hear the term coronary circulation, we're just talking about the blood that is servicing the heart itself. So like the right and the left coronary arteries, the circumflex, the anterior interventricular or left anterior descending, those were those coronary blood vessels. So this is what it looks like when a professional draws it. There you go. So this is the pulmonary circulation is when we're going from, let's start with systemic down here. Systemic. Now, this is also systemic up here. We're just separating the head region, everything above the diaphragm, from below the diaphragm. So this is systemic capillaries. These are systemic capillaries. And here we're receiving that all this blood is coming from the aorta into these capillary beds. And the oxygen goes into the tissues, picks up carbon dioxide. Here we go to the right side of the heart. We could have done the same on this other side, the aorta to the lower part of the body, to drop off oxygen, pick up carbon dioxide, and then end up into from the inferior vena cava into this right atrium. Then the blood goes into the right ventricle. Then it's pumped through this pulmonary valve. By the way, here's the tricuspid valve, back and forth. Pumped through the pulmonary valve, and it's either going to go to the left lung or the blood is going to go to the right lung to drop off the carbon dioxide, pick up oxygen, and then these are the 4 pulmonary veins. 1, 2, 3, whoops, this one and this one. 4 pulmonary veins are entering into this left atrium, and then the blood goes through the mitral valve into the left ventricle, and out it's going to go through the aorta, and the cycle just continues on and on and on and on. Obviously, if we're just taking blood from our heart laterally to our lungs, this is going to be a low pressure system. When you measure blood pressure, and we get to blood pressure in the next chapter, we're going to see how the pulmonary circuit, called it in the last slide, pulmonary circulation, pulmonary circuit, whatever you want to say, it's low pressure because we don't have to have a lot of pressure to send blood straight to our lungs, but we have to provide quite a bit of pressure to all of the blood that's going through the systemic circulation. So systemic circulation is a high pressure circuit. Interestingly enough, if we're sending, if our cardiac output, if we've got 5 to 6 liters, let's say 6 liters per minute of blood is going through the right side of the heart, then we have to have 6 liters of blood per minute going through the left side of the heart. And that seems a little outrageous because your body's huge and then your lungs are just these 2 organs, but both sides, the right and the left side, always remember, are pumping the same amount of blood. If one is pumping more than the other, we're going to have a backup somewhere. And so they're pumping equal volumes of blood. Now, this is just another view of the pulmonary circuit. And it's good to see this because here we have this pulmonary trunk and we're going into the left pulmonary artery or the right pulmonary artery. Arteries lead away from the heart, but they're colored blue in this picture because we're in pulmonary circulation. In the next chapter, when we get into blood vessels and systemic circulation, all arteries are red, all veins are blue. In the pulmonary circuit, they're exactly opposite. So we go to the capillary beds, and out here in the stroma, the balance of the lungs where we have the air sacs, that's where the capillary beds are going to be. And so we always go from an artery into a capillary bed, into veins, and back to the heart. And so here in this pulmonary circuit, we're going to have the pulmonary arteries that are blue that you see there, going into the microscopic regions around the alveoli and the capillary beds. That's where those are located. They're going to pick up oxygen, drop off carbon dioxide, and then return to the heart through the pulmonary veins, which you see in this picture as red. Don't forget that. This is another great opportunity to look at the serous membranes. This is a serous membrane. Looks, yes, blue. That is the parietal, meaning it's lining the body cavity. That's what the word parietal is when we're talking about the serous membranes, those membranes that are made to reduce friction. This is the parietal pleura. The second name is like where it is in the body. And here's the double part of that right there. That is the visceral pleura. And between these two areas, we have serous fluid with the viscosity slightly less of a raw egg white. Slickety. It's slickety fluid produced by these two membranes. And so when you're breathing, you cannot feel yourself breathing if your serous membranes are doing the right job. Now, around the heart, I have a hard time seeing what's going on here, but the blue serous membrane, that is going to be the parietal. It'll be on the outside of that. It'll be on the inside of the fibrous pericardium. This is going to be the parietal pericardium. Notice how the second word changed. Up here, we're around the lungs, so it's the parietal pleura. Here, we're around the heart, so it's the parietal pericardium, around heart tissue. And then if we are on the outside of the heart itself, that layer is the visceral pericardium. I don't know how many times I can say that and it not sink in. People have a hard time with that. Okay. Some points to notice. Here are the bronchi. These are the air tubes going into the lungs. And if we breathe in, the air is going to go through our mouth, down our esophagus, into the trachea, and then down through these bronchi. So what you don't see superior to this cut is the trachea, which will be right here, the windpipe, and then this is the esophagus behind it. Just for reference, look at your that is one of your vertebrae, and we have the ribs going around. There's the sternum in the front. Now let's talk about our heart valves. And I think I heard a car pull up, so give me a second to pause. Okay, so the heart valves themselves, remember we're going to have two different types of heart valves, 4 heart valves in all. The one set, they're called the semilunar valves, and these guys are at the base of the great vessels that are leaving the ventricles. In other words, one is at the base of the pulmonary trunk. It's called the pulmonary valve. The other is the aortic valve, and it's at the base of the aorta. And so if you're thinking of, I like to think of, just pretend like the ventricle is not connected to anything else but that big old aorta. There is a valve right here, which actually has 3 cusps. And there's always going to be blood in here. And when this ventricle is going to contract, it has to overcome the pressure that blood is forcing on the aorta walls. So while this ventricle is relaxing, the pressure that this blood is, it's just pushing down on these valves, keeping them closed. They're just little flaps, nothing more than that. They don't have a brain, remember. So when the ventricle originally contracted, it shot blood up. Most of the blood went forward, but some came back down and closed those aortic valves. So when the ventricle is at rest, the term for that is ventricular diastole. That's the adjective form of ventricle. When we're in ventricular diastole, the amount of blood that's just resting up here is pressing on the walls of the aorta. That's hydrostatic pressure, but we call it blood pressure, right? And so when the ventricle is at relax, the question is how much pressure is in this aorta from the blood that's just sitting there pushing up against the walls. That you can measure, and it's called diastolic blood pressure. Your diastolic blood pressure is the pressure experienced by the aorta when the left ventricle is in diastole. So say you take your blood pressure and your blood pressure is 120/70 millimeters mercury. This is the pressure part. If the blood here in here is pressing against the wall with the force of 70 millimeters mercury, that is your diastolic pressure. Well, when the ventricle contracts, that's called ventricular systole. It has to overcome this pressure right here to move this unit of blood forward, and the next unit of blood up. So when that left ventricle contracts with force, it has to contract with more force than 70 millimeters mercury of force. That's why diastolic pressure is kind of important, because it really determines how hard your left ventricle has to pump to get blood out of it. Now, imagine this left ventricle squeezes and has that squeezing, and we have enough force to open up this aortic valve here and move this pressure of blood on. When that is happening, this left ventricle here will have our little valve open, and the aorta here, and the left ventricle, pretend like it looks a lot smaller than that because it's really squeezing blood through. When it's squeezing blood through and the left ventricle is in systole, that means contraction, that blood is pressing on the walls of the aorta much more. And so that you can measure in a blood pressure cuff, and that is called your systolic blood pressure. And so your systolic blood pressure is a measure of how much pressure the aorta experiences during left ventricular systole. It's worthy of knowing, right? You're going to be a healthcare provider. You should understand what the difference between systolic and diastolic blood pressure is. Well, if these valves aren't competent, this is a very important valve. If it's not competent, the blood is always going to be, the left ventricle contracts, sends blood up, and then when it relaxes, the blood can fall back into the left ventricle. So we need these semilunar valves to be competent, to not prolapse, to not allow regurgitation of blood back into the ventricles. So as we said in the last lecture, these valves are responsible for one-way flow of blood through the heart. Okay, that was my cat coming back in if you heard the door open. Okay, they're responsible for one-way blood flow through the heart. That just startled me. I'm trying to get back to there. Now, the atrioventricular valves are for the same thing, for one-way flow of blood through the heart, except for the atrioventricular valves, as the name suggests, are between the atria and the ventricles. So these ones are very easily recognized because they're between the atria and the ventricles, and they also have the chordae tendineae attached to them, and the chordae tendineae are attached to papillary muscles, and so they're quite obvious when you're looking for them in a dissection otherwise. The one of the atrioventricular valves has 3 cusps and the other has 2. It's the one on the right side that has 3 cusps, hence the name, the tricuspid valve. It is between the right atrium and the right ventricle. The mitral valve, some people say it's the bicuspid valve because bi means 2. The mitral valve is on the left side of the heart between the left atrium and the left ventricle. So what these guys are doing, if we look at the AV versus semilunar valves, first of all, notice that the AV valves have the chordae tendineae, these tendinous strings, tendinous cords, attached to the papillary muscles. So you can see that. Notice that the semilunar valves, here's one, and here's another one, they do not have chordae tendineae because they work in a different way. The back pressure of blood coming back this way is what closes them. Now, what closes these valves, the right and the left, is also pressure. When blood is filling up in this, that is a massively large right ventricle, and then if blood is filling up in this left ventricle, pressure is building up, and that pressure causes those valves to go from the open position to the closed position. But then that's just during ventricular diastole, when the ventricles are relaxing and the blood is filling into them. But as soon as those ventricles contract, then they're going to apply a lot of blood force to that blood, and we want the blood in this right ventricle to go this way. We don't want it to go back up into the right atrium. So that's what the chordae tendineae and the papillary muscles are for. We have the valves coming into a closed position just as blood starts filling up in the ventricle. But as soon as it contracts, these little papillary muscles will pull on those chordae tendineae to make sure that while the valves are closed, they don't get inverted back up into the atria and blood doesn't flow backwards. So they stabilize those valves. All right, there we go. That's the difference between the two. Oh, no, I don't want to do that yet. We'll come back to that. Let's look at this cardiac histology. Remember that our cardiac muscle underneath the microscope looks like this. It's striated. The cells are short, fat, and branching, and we see a lot of the intercalated discs in there. These intercalated discs are regions like right here where two cells come together, and they pick up a lot of stain because at these regions, the two cells that are coming together, they have to be connected by desmosomes. You can call them adhering junctions. And what those do, what a desmosome does, is a special cell-to-cell contact. If the two cells are going to pull apart, they don't pull apart because they're full of desmosomes. So I don't really see that the artist has hit that in this picture. I don't see any desmosomes, but they're proteins, and so they stain darker, and that stain is picked up as the intercalated disc. Not only do we have desmosomes that prevent the cells from being ripped apart when each cardiac myocyte, or just cardiomyocyte, each heart cell, that's what that says. When each heart cell contracts, unlike skeletal muscle, when you have skeletal muscle is going to span a joint. And when it gets short, fat, and bunchy, it's going to pull on the bone that's on the other side of that joint. So we have that tension, that muscle gets short, fat, and bunchy. Well, these cardiac muscle cells, or the myocytes, don't attach to bones, they attach to each other. So when each cell gets short, fat, and bunchy, you don't want them to pull apart. That's the job of the desmosomes. But at the same time, pretend like I'm actually drawing two nice cardiac myocytes here. We have other proteins that don't look anything like this, but I draw them like little straws that connect the cytoplasm of one cell to the cytoplasm of another. That's called a gap junction. And the gap junctions are special cell-to-cell junctions that allow communication between two cells. If something is happening in one cardiac myocyte, it's happening in all of them because they're connected by these gap junctions. And that's what you're seeing in these intercalated discs here, these darker staining regions. Here, these striations are caused by the myofibrils, just like when we learned skeletal muscle, how the myofibrils are made of sarcomeres, and we have these, so they stain in a striated pattern. But the intercalated disc are due to these extra proteins. Notice how many mitochondria you have in there. These are the myofibrils, and the myofibrils are the organelles that are made of repeated sarcomeres, so they can all shorten and get fat and bunchy. That's about it. Now, between cells, there is more space between cells, between the cardiac cells than, say, skeletal muscle sometimes. And that is that fibrous skeleton of the heart that helps if the heart is going to expand. Part of it helps it to recoil. Okay, that's the end of this lecture, and we get into actual cardiac physiology in the next unit. So I would highly suggest before you watch the lectures that you review the action potentials that occur between along skeletal muscle cell sarcolemmas and along the plasma membrane of neurons. That's it for today.